All right, so Ab-Initio protein structure predictions. So this is the classic problem of given an amino acid sequence, we want to find a three-dimensional structure. All right, so that's the classic protein folding problems. This has been thought about ever since we really had a molecular understanding of the central dogma of biology, that DNA codes for protein, or for DNA codes for RNA codes for protein, and then that protein is going to fold into a sequence. So this has been thought about for a long time. We talked a little bit about protein design. This is the opposite of the protein design problem, where we want to make a structure and we have to figure out the sequence. This is what nature's already set up for us. We have a sequence, we'll figure out what structures they make. And you could ask why even do this, because we have a lot of known structures now, and we also can do some design, and some people with the Structural Genomics Initiative, there are tons of structures coming online all the time. Well, you still might want to do this for brand new sequences where we don't know the answer, but beyond that, this tells us something about protein folding. What are the rules to go from sequence to structure, and do we understand the rules? And it tells us about the physical chemistry. So this is a very rigorous test, really, of our biophysics knowledge. Do we understand how proteins work? So that's a big motivation. So there's two big concepts underlying the protein structure prediction approaches that I want to talk about, or at least the problems to think about. One is Levinthal's paradox. So Levinthal, back in 1969, threw out the question of how does a protein know how to fold. Okay, so this is now we're talking about the what do we know about protein folding, and how this problem informs that. So what do we know about protein folding? So Levinthal estimated that well, the average human protein is about 300 amino acids, which is quite a bit. And he said, well, what if each residue could be in one of three conformations? It could be either alpha, or it could be beta, or it could be something else, which we'll call Luke, random foil. So let's say there were three states for each. Three states possible for each amino acid. How many conformations are there available to the protein? That's the question. So let's say we had 100 residue protein. And each one had three potential states. How many states are possible for the protein as a whole? Jessica.
3 to the 100?
Yeah, so the first residue could have three states, the second residue could have three states, the third residue n times, so we'll get 3 to the number of residues conformations, or in this particular case, we'd have 3 to the 100. 3 to the 100 is a pretty big number. Let me say 3 to the 300. I said the average human protein is about 300 residues long, so if we go 3 to the 300, this comes out to be 10 to the 143. That's a pretty large number. To put it in perspective, the number of particles in the universe is about 10 to the 80. So this is ridiculously large. This is much larger than the number of particles in the universe. And this is a pretty lousy estimate, too. This is saying each residue can only go into one of 3 states, which really, you've looked at protein structures, you can change phi, psi, chi angles. There's more than 3 states per residue, right? So just saying it's alpha or beta is really not specifying what kind of alpha it is. So this is actually a horrible underestimate. So the paradox is, how do proteins find the folded state when the conformation is so large, when they have so much conformational space to search through? That's Levinthal's paradox. This is a paradox for protein folding, but it has huge implications on our efforts to find the fold, because in the computer, we're up against that same kind of exponentially large conformational space. Okay, so the second core concept, so there's Levinthal's paradox, the second core guiding idea is Anfinson's dogma, and this came about, well actually Anfinson got the Nobel Prize in 1971 for this work. So Anfinson asked the question, what is it that controls the protein fold? You could imagine we knew some things about translation happening in the cell and that the RNA gets translated into protein and does the RNA and the machinery in the cell, the ribosome and the Golgi, does it put the protein into the shape it's in, or does the protein know how to fold itself? And what Anfinson said is that all information for protein folding is contained in the amino acid sequence. Okay, so and what Anfinson did is he just took an enzyme and he heated it up, or he chemically denatured it, found that the enzyme fell apart, so it was unfolded, and he reversed the conditions, brought the temperature back down, changed the condition of the solvent, got rid of the denaturant, and the proteins refolded, and then the proteins were active again. And so what that tells you, if you can do that in vitro, it tells you the cell machinery doesn't matter. It doesn't matter that it came off the ribosome one at a time. It doesn't matter that it got exported through certain channels or pathways or anything like that. So that tells you it's a purely physical phenomena that the protein molecule itself knows how to fold. Okay, is that true? So is his dogma true? That's a question for you. Is this true or not? I see heads shaking. Yes, I see heads shaking no. What do you think? Do you know of counterexamples? Yeah?
Like there's proteins that need chaperones.
Some proteins need chaperones. Why do proteins need chaperones?
To get the right sort of environment in which to fold.
And yeah, so some proteins can misfold. The chaperones, some of them look like big containers. They have hydrophilic walls that helps turn the folds inside out and then refold them the right way in. So some of them do get stuck and need some refolding. Others? You have other examples? Yeah, sure.
There are proteins with the same amino acid sequence known to fold differently with synonymous codons. So if the translation rates for some codons are faster than the other, they could be stuck in local.
So that tells you that something about the rate of coming off the ribosome is controlling the folding. Interesting, yeah. Other proteins can get kinetically trapped. So thrombin folds and then it gets cleaved, and once it gets cleaved, that particular state it's in is no longer what it would be in if the cleaved parts had folded on their own. So sometimes chemical reactions, protease, cutting the peptide bond can stick things in a state that is not the lowest energy state. Okay, so there's a few ways around this. But by and large, this holds for most small globular proteins. This is a little bit of a reflexive definition because proteins will be small and globular if this is reversible. So we call these thermodynamic proteins. Thermodynamic proteins or biophysical proteins. These are proteins we'd like to fold in the computer because we know they can do it in vitro. We don't have to have special kinetics or other things. So what does this tell us? This tells us that all we need to know is the thermodynamics of the particular molecule, that we're going to look for the lowest energy state, that we don't necessarily have to follow the path that the cell used to create the structure. Okay, so that's pretty key for us to do what we want. Okay, so from these two ideas, there are a couple pieces that we need for a folding algorithm. Folding algorithm requires Number one, thinking about Levinthal's paradox, the first thing we need is a search strategy. And what this means is we're looking for an algorithm that can create many candidate protein structures. An algorithm to create candidate protein structures, or the jargon we use in the field is we call these decoys. Each candidate structure is a decoy, which might be the right structure, might not be. It's a set of structures that we make in looking for the correct structure. The second thing we need, once we generate a bunch of structures, if we can overcome Levinthal's paradox and generate many, many structures, then the next thing we need to do is tell which one is the correct structure. And this is coming back to Anfinson. Anfinson says you're looking for the lowest, and it's a thermodynamic process, so we're looking for the lowest free energy state, and that lowest free energy state is what it would fold back to if you were to nature it and then come back to it by logical conditions. So the second thing we need is a scoring function. And this is to discriminate near native structures from all others. So let's say we overcome Levinthal, we make 10 to the 143 structures. We want to go through each, give each a score, which really we were looking for some kind of energy, and we hope that the lowest, what a good algorithm would do, a good scoring function would do, is the lowest energy, the lowest scoring structures. Those are going to be close to native, or even the native structure, because that's what nature does in actually folding things. It finds the low energy structure, so that's what we're looking for too. All right. All right, so what we're going to look at today is Kim Simon's, David Baker approaches that were developed in 1990, or published anyway, in 1997 and 1999. I posted both of the relevant articles on the website, and you can read through, and there's two main concepts in these papers. The first paper, the main concept is that local sequence gives a bias toward a small number of local conformations. And we'll go through what this means in detail. But we're talking about a local sequence, so maybe just 9 amino acids. Those will have some biases towards some local conformations, some small number of conformations that those could take on. And then second, that the non-local interactions, so by local that means along a subsequence of the protein, so non-local meaning far away in sequence, say one helix down a turn and back around as things are interacting. The non-local interactions are going to preferentially stabilize the native-like global structure. Okay, so what Tim Simons and David Baker did is they broke this down and said we're going to look at local conformational preferences, and then and that'll be set by the local amino acid sequence, and then the global amino acid sequence will be stabilized drawing from these local ensembles versus optimization. So you've heard of molecular dynamics, and you might have taken statistical mechanics, and in statistical mechanics, we think about the molecule, we make an ensemble of all of the possible conformations of the molecule. You can kind of picture that in your head, where all the different shapes we can fold the protein into. In molecular dynamics, you're looking to characterize that whole ensemble. You want to know every single state and assign all the energies to all of those states. Okay, so the idea is to capture the thermodynamic ensemble and understand the ensemble. The reason being is that you would be looking for, so an ensemble approach and find average thermodynamic properties for example, if you wanted to know the density of air in the room, you might take all of the configurations, some of which the molecules are over here, over there, or denser or less, and then you'd average them all together to get an average property, an average heat capacity, or an average viscosity, or something like that. So in an average property, you have some property A, we'd take an integral or a sum over the property A for some configuration times the probability of seeing that configuration in an ensemble, and we'd sum over the whole ensemble or integrate over the whole ensemble or the whole conformation of space. So if we want to know the free energy of a protein at a particular set of conditions, you would want to sum over all of the high energy and low energy configurations to get an average energy of the whole ensemble. That's not what we want. So what we actually want in protein structure prediction, I don't care about all of the states, I care about the one state that dominates the ensemble, which is the folded protein conformation. Okay, so this is one of the magic properties of proteins. Unlike general polymers that might be more like spaghetti or even some disordered proteins, folded thermodynamic biophysical proteins fold into a three-dimensional structure. That's the one structure we're looking for. So instead, what we're looking for is the optimization, the lowest free energy. So our optimization approach seeks the one lowest energy conformation. So we want to minimize the energy as a function of whatever that conformation space X is, wherever those coordinates are. Okay, so it's not actually necessary for us to sample everything, which is a good thing, because I don't have 10 to the 143rd conformations that I want to enumerate, but I don't need to. If I know a lot of those are irrelevant, the energy is too high, they're not the lowest energy state, I just want to seek the lowest energy state. So as soon as I see one structure that's lower energy than another, I might be able to throw out the higher energy state and not worry about the memory there. Whereas in molecular dynamics, you're going to keep track of all of that, or on Monte Carlo, when you're trying to capture the ensemble, you're going to do statistics on the whole thing and capture all of that. You'll have a lot more information, you'll have thermodynamic information, but it comes at a slightly higher cost. Monte Carlo folding algorithm. And this is from Kim Simon's paper, 1997. And the reason I start with these papers, I think we mentioned before the CASP challenges where there's blind predictions of protein structures. So in 1997, that was when this was developed, and shortly after that were the first CASP challenges where there were correct protein structures that came out of these blind challenges. Monte Carlo folding algorithm is going to be based on what we call its fragment insertions. So the idea is that we were going to take the protein sequence, so this is the protein sequence, and all along that sequence we have phi, psi, omega angles on the backbone, phi, psi, omega, phi, psi, omega, phi, psi, omega. The idea is we will take some random point in that sequence. We will take the amino acid sequence there, and we'll think about what is that amino acid sequence, what would it like to be? Would it like to be a helix? Would it like to be a strand? Would it like to be a turn? So we'll take these 9 amino acids, and maybe we'll say it should look like a turn, something like that. And then we're going to take, so we'll start with the protein being straight. So phi and psi, omega might be all 180 degrees, all nice and somewhat straight. Then we would take a small piece of structure out of a pre-calculated library of small local configurations. And then we're going to take these angles and we're going to paste them in. We'll cut out this section and paste them in. So if we do that, this would come along here, and now we would go like that from here to here, and then the rest of the protein would head out that way. Does that make sense? So I'm going to take a fragment of a protein structure, I'm going to paste it in. This is a 9 amino acid part, and that's what I call a fragment insertion. I'm inserting a fragment of structure into the protein configuration. That's the basic idea of the Monte Carlo folding algorithms that Kim Simon's put together. Then once we do that once, we'll repeat. We'll do it again. We'll pick a different segment, and then we'll put a folded piece in there, and then we'll take a different segment and put a folded piece in there, and we'll do it again and again and again. So we're swapping on a folding algorithm. There's a straight protein. That's the final folded configuration that came out in a crystal structure. So that's throwing a few fragments in, and immediately you get some secondary structure. You see a helix pop out, and then we see, again, we're flopping in major pieces of amino acid fragments, and you can see those strands flopping all around. Eventually it starts to stabilize a core, and now we see 3 strands in a helix, and we've got this 4th strand that then continues to flop around. And most of the places has a fairly lousy energy, but when it finally pops in, that'll really set the energy and give us a nice low minimum energy, and now it's fairly close to that native structure. So that's the idea of that's what the folding algorithm is doing. Okay, and then this is smaller fragments. These are 3-mer fragments being inserted, 3 amino acids at a time. It's getting smaller, jiggling around. You've probably heard Monte Carlo before. Monte Carlo is a pretty standard optimization technique that's used in a lot of different for a lot of different problems. In general, Monte Carlo starts with making some kind of random move. You start with some kind of state of your system, in this case the stretched out protein. Then we do some kind of random move to generate a test configuration. Okay, so you have some initial start configuration, then you throw in a fragment and you generate a test configuration. So in this case, we pick a random move, we pick a random position in the sequence, and then we pick a random fragment. So this method, one of the nice things about this method is you pick these random things that you don't necessarily have to know where to go, you just try random things and you see what works. So it's a stochastic algorithm. So we do a random move, and the next step is we evaluate the change in the energy equals the energy of the test configuration minus the energy of the old or the start configuration. Okay. So we see after we put in that fragment, did the energy go up or did the energy go down? That's the question. And based on that, we decide what to do with that test configuration. So again, remember we're looking for the lowest energy state, so I say if delta energy goes down, then we accept the test configuration, test confirmation, and that becomes our new starting configuration for the next round. So that's easy. If the energy goes down, that's good, I want that. If the energy goes up, we don't just necessarily reject it. What we're trying to do, and where this comes from, is from those ensemble theories of coming up with what's a thermodynamic ensemble of the high energy and low energy positions. So to do that, what we do is we generate a random number. So that random number R will be between 0 and 1. Based on that random number, we're going to decide whether or not to keep the configuration. So if the random number is less than the exponential of opposite delta E over KT, then we accept. Otherwise, we reject the test configuration. Okay. So this expression I kind of threw out of nowhere, but this comes from statistical mechanics. This has to do with the probability of seeing one state relative to another in a thermodynamic temperature equilibrated ensemble. So higher energy configurations you're less likely to see, and lower energy configurations you're more likely to see, and it depends on the temperature of the system. So this is Boltzmann's constant K and the thermodynamic temperature. In the computer, that's a variable we'll be able to set. Then what else? Then there's some bookkeeping, which can consist of a variety of things, but one thing for us is we're looking for the minimum energy configuration. Yeah,
So when you generate R, do you draw it out of like a uniform distribution?
this is a uniform distribution from 0 to 1, that's right. Yeah, so this e to the minus e over kt tells you the percent or the fraction of time that we should be, or 1 minus the fraction of time that we should accept the configuration. Okay, so the bookkeeping, since we're looking for the minimum energy, we want to keep track of the minimum energy configuration we see. So if the energy of the test configuration is less than the energy minimum that we've ever seen during the whole run, then we are going to store the confirmation, the minimum confirmation. This would be our low energy configuration, so that later we can pull that out. There's more bookkeeping, but we'll keep it like that for now. And then after that, we repeat. So then we go back. We generate in a new fragment, see if the energy goes down. If the energy goes up, take a random number, maybe keep the configuration, and then repeat. So we put in a fragment, test it, put in a fragment, test it, just go back and forth. And that's what you saw in the video. Okay. All right, so what's happening conceptually? If we think of an energy surface, we think of the x-axis here as different conformation spaces. You move the protein around. As you move the protein around, the energy changes. So maybe we start here in conformation space, and then we throw in a fragment. That might move us to here. One of the nice things, if we were at MD, we would have had to continuously move through configuration space and follow the motion of the molecule. We don't have to do that here. So one of the advantages of MC is we get to jump in configuration space. 9 pairs of phi psi angles we replace at a time and go somewhere else. We look at the difference in energy. In this case, the energy went down, so we'll keep that configuration. Next move, we end up here. The energy goes down again, we keep that. Notice how we jumped over a peak here, that's kind of nice. The next move brought us up here, the energy would go up. We would then take this delta E, we'd divide by KT, and we'd use that to see, take the opposite of that, the exponential, we use that to see if we accept that position or not. So maybe this time we accept it, and then maybe that next time that lets us move over here. Because you can move uphill, you actually can climb up mountains with slow steps, as long as you get good random numbers. But you also should be, it should be really easy to fall into these minima so that you can sample near the minimum of these energy surfaces. Okay. The board is small. I'd like to keep this up, but I think I'm going to erase. So we have this. Do we have any questions about the general algorithm? So this is the general idea, and this is Monte Carlo. Any kind of Monte Carlo is like this, where there's a trial move, there's a state of the system, a trial move, a test based on the Boltzmann factor, and then you go back and optimize that. If you do this test right, this expression gives you something called detailed balance, meaning the states going one way is the same as the probability of states going the other way. If you do that long enough, the whole system comes to equilibrium. So if you keep statistics, you can get equilibrium statistics out of this. But we're not going to do that because we're looking for that low energy state. Okay, so trick number one that Simons and Baker did. One is, the multi-start trick. So the idea here, so this is an optimization calculation. So there's a good parallel to this. If you wanted to find the highest elevation on Earth, and you did a Monte Carlo search, so you became the agent, and we're here in Baltimore at, I don't know, an elevation here is maybe 200, 250 feet. And if you just started wandering about, and you tried to wander downhill, but every now and then you drank some coffee and went uphill, and you're actually, no, let's go the other way. You try to walk uphill, and then every now and then you get lazy and walk downhill, and you allow some of those. Could you wander, and could you find the highest point on Earth, is the question. So maybe if you wander around, maybe you find Mount Mitchell, which is our largest peak in Maryland. Or sorry, Mount Mitchell is the largest peak in the Appalachians. And, but you're probably not going to get all the way over to Mount Whitney in California in the continental US, right? You're probably not going to get to Mount McKinley. So what do we do instead? Instead of just having one hiker moving around looking for the summit, you deploy a bunch of hikers. So you could start, we could take the whole class and go out and wander, and now maybe we'll find the Appalachians faster than if it was just one of us. But we can also start each hiker at a different place. So what if we start with a parachute, we pick a random latitude and longitude, correct for the sign with the longitude, and then we drop people around the Earth. And maybe some people would be dropped in California, and somebody would get up to Mount Whitney, and maybe we'd even have somebody in the Himalayas we found. We'd find Mount Everest, right? So that's the idea, is the multi-start, is we're going to start multiple searches from different places in configuration space. So we're going to restart from new random state with a new random trajectory. So the idea is that restarting is more efficient than waiting for one trajectory. This is more efficient than following single trajectory. So partly just because you have more things going on, if we had more hikers, that's good, but also because you don't get stuck in these local maximums. So if you start wandering around the Appalachians, you think you're doing pretty well because you're going uphill, but you're not even half the elevation of the Rockies, and you're not even half the elevation of the Himalayas. So you really need a global search to lock things in. So that's trick number one, is that, so we get stuck in local minima. Trick number two, it's a little more technical, is called simulated annealing. So the idea behind simulated annealing is we are going to adjust the temperature. So a big factor in this Monte Carlo search is the delta E over KT. That tells us whether we keep new configurations or not. So what happens if I have a high temperature? At high temperature, delta E over KT is going to be small. Minus E to a small number is close to 1, right? Because E to the 0 would be 1, so R is less than 1, which is almost all the time. So if temperature gets really large, we'll accept every move. So at high temperature, you accept every move, you move all around. It's like there's no energy surface at all. In fact, another way to look at this is take the energy surface and squish it. A raising temperature would squish it so it looks flat in a limit of infinite temperature. So you can move anywhere. Okay, so high T allows large steps, which means we can move fast in configuration, in conformation space. How about the opposite? What if we send T colder and colder? If T is very small, delta E over T would be large, so E to the minus a large number is going to be close to 0. There's going to be a very small chance of accepting a move, maybe 0 of accepting an uphill move. If we go to 0, there would be no chance. We'd only go down the hill. So at low T, we force trajectory toward low energy basins. So that's good because we're getting low, but it means we'll never get over a barrier to see the other side of the barrier. So you actually want both of these in a search. You want to search broad and wide, but then you want to go down low. So how do we do that? Well, we simply, through each of our steps, we change the temperature. So we'll start out high, and then we'll decrease the temperature as we go. Okay. So what Kim Simons did is there's 10,000 fragment insertions in the algorithm. Temperature starts real high at 2,500. This is KT actually, and it goes down to 10. It's not really meaningful until you know the units of the energy, but in any case, it starts. It changes 2 orders of magnitude. And so that's a big, more than 2 orders of magnitude, that's a big change. It's moving around a lot, and then toward later it moves around very little. And if you were paying close attention to the video, you would have seen that. It makes crazier moves in the beginning and less crazy moves at the end. All right, so I told you there was extra bookkeeping. One of the extra bookkeepings is adjust the temperature every time around. Definitely one of the core key ideas that was novel about this method when it came out. In fact, a lot of people in the CASP challenge call this cheating. That's how blatant it was. Because really what you're doing is stealing protein structures and throwing them in. So I said, well, you're not really calculating it and driving it from the get-go. You're not doing that ab-initio. That's not ab-initio folding. That's threading or fold recognition or something. But it's only doing 9 residues at a time, and you're just trying to recognize what it does. But so there's some legitimacy to those criticisms. This is different than people that start with Newton's equations and try to come up with the right answer. But in the late 90s and the early aughts, you couldn't do that with molecular mechanics potentials. They really didn't even have the property that the low energy state was the native state. So it was necessary, and I found this is an interesting lesson for science, because when you can't do something that's really hard, one way about it is to cheat and break it up into smaller steps. Get some pieces you do know and see if you can get things to work, and then gradually go back and undo the stuff where you had to cheat. So, okay, so let's see. So the idea of protein fragments is that the sequence allows a relatively small number of local structures. So I'm going to show you what the fragments look like, and these fragments are 9mers. So we take 9 amino acids at a time as a piece of fragment, and they tried different lengths. There's a reason why 9 works. If you start with 1 amino acid in the middle, that means you're looking +4 and -4 along the sequence. That's about how long it takes to get around a helix. So it's about the local interaction length where hydrogen bonds and things matter. What we're going to do is have 25 fragments for each 9 residue window. Here's residues 1 through 9, 2 through 10, well, they're from different places, but here's different windows through the protein. And what's showing at the top is in the native structure. That's the conformation of the backbone, 9 residues. And here's 25 different conformations that are put in the library. So at some of these places, there's a lot of helical fragments in here. For this one, there's a lot of turn fragments in here. And the idea is you start with a half-decent set of fragments that has some diversity in it, and also focused a little bit at times toward what each of these 9 residue fragments might do. So how do you pick the fragments? We're going to take structures from 25 nearest sequence neighbors in the PDB. So we'll take that. So first of all, the windows are overlapping. So residues 1 through 9, 2 through 10, 3 through 11. So all those windows overlap. And for each window of 9 amino acids, we're going to take those 9 amino acids and ask in the protein data bank what other sets of 9 amino acids are similar to that? So there's probably not one match, right? Because there's 20 to the 9th different 9 residue sequences. So it's you're not going to have an exact match, but you might have some that are more similar than others, and that's what you're looking for. The sequence neighbor, so how do you tell how close a sequence neighbor is? Well, there's some sequence tricks, which I won't go into. If you've had bioinformatics classes, you can do that, but basically you're going to look at the amino acid, the sequence you have, look at profiles of what is similar to that based on substitutions. So you have a profile for your query sequence, and then every 9-mer sequence in the protein data bank, they made a profile for that too, and then you compare these profiles. So sequence neighbors determined by comparing profiles. So we look at probability of amino acid from alanine through valine, any of the 20 amino acids at each position I. So at position 1, maybe it is a valine, but in similar sequences that you find in BLAST, sometimes it's an isoleucine or sometimes it's a glycine. So you have a probability of substitution within your sequence, and then you match that profile to all of the profiles of the fragments, of the subsequences in the PDB. So the distance is we sum from residues 1 to 9, and we sum over all 20 amino acids. The sequence probability for our query sequence for amino acid at position I minus each position in the fragments from the protein data bank, and you sum those. So you sum over all 20 amino acids, the probability of seeing that amino acid in all 9 positions. And so we really haven't gone through all that math on analysis, but for those that do know, that's the detail. All right, last piece for the fragments is that the fragments are idealized. If you just throw in a fragment from the protein data bank, it's got a range of bond angles, bond distances, and as you've seen, those change a little bit. We've done that in the homework. We've looked at that in real structures. Just because a bond is stretched in one particular protein doesn't necessarily mean it's stretched in another protein. So what we do is we take the 9-mer fragment and idealize all the bond lengths to ideal angles and idealize all the bond angles. Now it's not going to match the configuration. Then you adjust the torsion angles to match the positions of the atoms as close as possible. And you can usually do that without adjusting it very much. So you change the torsions a little bit and keep the bond angles and lengths the same. That way, all of these fragments have ideal bond lengths and bond angles, and that makes it easier as you're substituting things in. So, bond lengths and angles are ideal. And with minimal root mean squared deviation to the experimental structure. In the paper, Kim Simon's paper says this avoids considerable inaccuracies. That's how they describe what happens if you don't look at a video. So I think that's an understatement that whacks things out a little bit. All right, so any questions about the set of fragments, about the Monte Carlo search algorithm? Okay, so now you know the general algorithm that we use to fold a protein from scratch. So that tells us about problem number 1, the Levinthal part and the search part. So we're getting around, so how do we get around Levinthal, the 10 to the 143 configurations? The fragments do it for us. The fragments tell us at each position there's only 25 things to search through. And we randomly pick one. We'll get another location, randomly pick another, and zip through those. So that goes really fast through configuration space, and it means that every local configuration, every 9 amino acids in a row, is consistent with what actually works. So you're not going to have any configurations where things completely clash, because all of these fragments are from real proteins, so they're not on top of each other. Now you might put one in and the rest of the protein will clash and it won't work, and then the energy will be ridiculously high and you'll throw that out, you'll try a new fragment. Yeah, Drew.
So these fragments are based on configuration, right?
What do you mean?
It's only in the when it searches the PDB, finding a very similar 9 residue sequence imply it's conserved. And
Well, okay, you might take it out. Okay, if you're looking for a particular kinase and there's another kinase that's a homolog, you don't want to fold de novo because you've already got a homolog to start with.
Okay.
So, yeah, you're right. If you just did this blindly, you'd pull out homologous pieces from homologous proteins, and it'd be kind of trivial. It wouldn't really be a blind search, right?
Yeah.
So this works even when things are not, so what you're looking for is distant relations. And when this algorithm was tested, what happens is for folding a particular protein where they know the answer, you throw out all the homologs. So if you're folding a particular kinase, you throw out all of the fragments from those homologs, generate your fragments without any near homologs. And so they're all distant relatives, and that's the only true blind test. Well, it's not, that's a little more of a fair test. That's actually tested by your algorithm. The real blind test is in the blind challenge where it's a CASP target that nobody knows the answer to, and you don't know. Then you'd use everything you can, right? But the tests in-house in validating the method are done by excluding the homologs from the fragments. Yeah. Other questions? Cool. Okay, so we can make a bunch of configurations. What else do we have to do?
I just have
Yeah.
one question. So we're still talking about a small globular protein. Okay.
Yeah, so if this were a 500 residue protein, it sounds a little crazy, right? Because a 500 residue, you often see a domain and another domain, another domain. If you have that whole thing flopping around, you're just going to make a lot of mess. So what you want to do, we'll talk about this a little later, if you wanted to say if you had EGFR and you didn't know the structure for that, the epidermal growth factor receptor has 4 giant lobes and they open and close. What you try to do from sequence is identify this is probably a compact piece, this is a compact piece, and you try to chop up the domains first, and then each single domain you try to fold. And a lot of times that'll work, a single domain you can fold as a biophysical protein. Yeah, but I think a large protein. So we'll talk about this, but in general, this works decently up to about 100 residues. 150 is starting to push it, 200 is getting large. So there are successes with larger proteins, but the search space becomes exponentially larger. So yeah. In fact, the Simon's paper, if you look at that, almost all the sequences are under 100. Some of them are in the 50 to 80 range. Okay, so what else do we have to do? We can make a bunch of structures. What's the other part? Emma? What else do I need for a folding routine once I have a bunch of structures? Tina? Bill?
Score.
You need scoring. Okay, so if there's 2 things we take out of the class, it's 1, you need to search all over the place smartly, and 2, you have to have a score, an energy function. Okay, so scoring. So we could just use, we've been talking about this for 3 lectures now, right? We could just throw in that energy function, except the fragments don't really have all of the atoms, right? It's just the backbone, and you've seen that everything depends on these atom distances, the solvation score, and the van der Waals. You really like all the atoms. You really haven't done everything. There's a reason, so we actually want to do something a little bit different for this folding. First of all, we don't really want the whole amino acid in all the atoms, because that's, I don't know where to put the side chain, so I want to avoid that problem for now. So I'm going to do a trick of multi-scale modeling. So we're going to make a representation of the protein that doesn't have all the atoms and doesn't ignore, but it represents the side chain in a simpler way. This is the centroid representation. That's a bit of Rosetta jargon. Rosetta calls it a centroid. In general, what this is coarse graining is what it is usually called in the modeling literature, where you take a bunch of atoms and you coarse grain them into something more united. And so we've actually seen this before in Neria, in the MD paper. They took the hydrogens and they plugged them into the carbons. So we had a CH3 atom where the hydrogens were a united atom. That's a coarse grain of those 4 atoms. So in Rosetta, we're going to have a nitrogen, an alpha carbon, a carbonyl. So Kim Simons wanted the whole backbone because, well, to do phi and psi, you've got to have those atoms, right? But then the side chain, you've often seen this written like this, where you put R as the side chain. This is the centroid representation. Now R is going to be an atom, just like C and CO, and the C and the C and the O and the N. This is an atom that is going to have a volume and a position. Okay. So what they've done is they tabulated positions for R, given the location of N, C alpha, C, and O. Let's label that C alpha, so that's separate from C. And then they tabulated parameters like the radius, or the distance between R and a nitrogen atom for calculating bumps. So would this R group overlap with a nitrogen in another amino acid over here? Well, how, if we could place this R at a certain spot, how far does that need to be from a nitrogen? And so it was real simple. It was going through all of the, basically took the side chain atoms and averaged the location, found the center of mass, and placed those relative to the backbone atoms. And then from there, looked at how far that was, say, from the nitrogen, and how far do you ever get, what's the closest you ever get? So we're allowed to go that close and no closer, and anything closer is a bump and won't be allowed. So that's the centroid representation, and it's very handy because it's going to be much faster. We're not going to have to do any chi angles. We've only got 1, 2, 3, 4, 5 atoms per residue. That's going to speed things up tremendously. But now the problem is, so we do that, but then you ask, this is similar to Myria's united atom, where CH3 is one thing instead of CH. Okay, so that's great, but now we just talked about all these energy functions from physics and physical principles about energies for atoms, but now this is an atom. So what does it mean to have a van der Waals radius for R? What is the solvation energy for R? What is the hydrogen bonding potential of R? Well, I don't even know where the donor and acceptor is on R. So I can't just plug in the physics-based energy function. So what, in 1999, what Kim Simons did in the second paper is talk about a scoring function development. And what he did is come about it from a different way. So we talked about in the hydrogen bonding function how we could do this from statistics. We could take a physical idea, look at statistics and parameterize it. They did the same thing except not necessarily with just hydrogen bonds, but they started to think about, well, what are the different things that we could use to describe good structures and bad structures. So this is where we start to use the word score more than we use energy, because we're looking to score what's better and worse. So this is about knowledge-based scoring function development. So we need to make a scoring function, and again, what they did, kind of like with the fragments, they said, you know what, there's a whole bunch of structures in the protein data bank, let's use those. Since molecular dynamics seems to be failing at line structure prediction, let's use what we know about proteins and see if we can at least imitate what nature does. So let's use that database. So let's use the database of structures and come up with some knowledge to recognize a good fold from a bad fold. So this is the process of how you do this. Identify features that may contribute to distinguishing proper folds from non-native folds. So you could start with that Monte Carlo algorithm, generate a bunch of configurations, and then look at them and say, which of these look like a protein, and just use your intuition. can I tell what things look like a protein, what things don't look like a protein? So if it generates something and it's all sprawled out, that doesn't really look like a protein. Protein is usually compact. If it generates something and it's got a bunch of aspartic acids all together, well, actually that might happen because maybe there'd be an ion. Say a bunch of lysines all together, that might not happen because it's too much positive charge in one. Well, that still could happen because maybe it's binding DNA. But if you look for something that maybe wouldn't happen, a bunch of hydrophilic right next to a hydrophobic, a charged ion surrounded by hydrophobics, you'd never see that in proteins. So you're looking for features that wouldn't happen in proteins that you can distinguish good folds from bad folds. Second, we're going to quantify this by choosing variables to describe the feature, and then estimate probability densities. So in the hydrogen bond, you'd say, well, this hydrogen bond has a certain distance, so let's use the distance as the variable, and we'll tabulate and we'll make a histogram. And then step 3, we want to evaluate the effectiveness alone and in combination. So the feature might be that you have to have good hydrogen bonds, but that's probably not enough. So you might need to combine this with other things. How many people have heard of Bayes' theorem? A couple. Okay, so Bayes' theorem looks like this. So we're getting away from physics a little bit. We talked about physics the last week and a half. Now we're going to get towards statistics. So the collaborator on this, there are a couple of statisticians. One is Ingo Roshinsky, who's here at Johns Hopkins down at the statistics department, the biostats department. And what Ingo contributed was some of this, this formulation. So let's see, probability of two things happening. Probability of A and B happening. So this would be, say we do some random things. Roll a dice and flip a coin. What's the problem? We get a 3 on the dice and heads. Two things happening. Well, Bayes said you can do some conditional property based on this. Probability of A given B times probability of B. So if the two things are completely independent, like dice and flipping a coin, then those are, you can just multiply the two probabilities together, but maybe they're related. What's the probability the temperature bypasses 75 degrees today and that it rains? So you could separate it and say, what's the probability it gets past 75 given that it rains times the probability that it rains at all. So you could separate it that way. You could also write it the other way because A and B are equivalent. You could say, what's the probability of B given A times the probability of A. So now I'd say, what's the probability to get it to be 75 degrees? And if it does, what's the probability of it raining given that it's 75, it's already past 75 degrees? Okay, so we can just say we can separate out two things that might be related into some different pieces. Okay, so that's just a simple conditional probability law. What Bayes did is rearrange this. So Bayes said A given B. We're going to flip B over to this side of the equation. There's P B given A times P A divided by P B. So if we wanted to know the probability that it gets, I forget which way I said this now, the probability that it rains given that it's 75 degrees is the probability that it's 75 degrees given that it rains times the probability that it rains divided by the probability that it is 75 degrees. So you can relate the two independent phenomena and the two conditional phenomena together. Okay, so that's Bayes' law. The 99 team said, we want to know the probability of having a certain structure given a sequence. So that Monte Carlo algorithm generates a bunch of structures, and we want to know what's the probability that one of those structures is right. We have the sequence, so what's the probability that that particular structure is right, and then we'll compare the probabilities of each one, and we'll see which one we think is most likely. So this is the statistical way of thinking about it. The energetic way would be thinking what's the lowest energy. It's the same as saying what's the greatest probability. So the energy and the probability, they're related through a natural law. We'll get to that later, but we're looking for the maximum probability of a structure. So what's fun about this is Bayes lets us flip this around. The problem is, I don't know we really don't know the physics, especially with the centroid. What's the probability the structure's right? I don't know. I don't know that energy function. But if I can flip this around, maybe I can put it in terms that I do know. So we change this into probability of the sequence given the structure times the probability of the structure divided by probability of the sequence. Okay, so now let's think about each of these. Probability of the structure. So I have the Monte Carlo routine, and it might generate some configurations. And I'd say which of those structures, structure 1, 2, or 3, is most likely to be correct. I don't know. But I could say, given structure number 1, what's the probability that the amino acid sequence fits on that structure? So here's a structure, does the amino acid fit on that structure? That might be something I can ask. So that's what term 1 is about. Term 2, probability of the structure alone. So just given these coordinates and nothing about the sequence, is that a good structure or not? And on the bottom, probability of getting the sequence at all. And then I'm going to compare these together. So when I compare these, probably the sequence is the same for each of these. So this is actually going to be a constant. If I'm looking for the maximum probability, I want the maximum that the sequence will fit on the structure times the maximum of the structure being correct. Okay, so for example, given this structure, if I put the sequence on, if all those hydrophobics are in the middle, that might be a good sequence for that structure. If the hydrophobics end up on the surface, that might be a bad sequence for that structure, right? So you might be able to guess what a good sequence is for that structure. And same thing with the structure alone. If I had a structure that looks like this, that doesn't look very compact, that doesn't look protein-like, it's probably not the right thing. Although there are disordered proteins, but we're going to ignore those for the moment. Do you have a question, Shoya?
No, it was related to this only. So the probability of structure is the probability based on the knowledge through our observation of the PDB, or is there an energetic term also?
That's exactly right. The probability of seeing a structure is based on features from the protein data bank. And we'll look at that. So we'll say, are the helices crossing in the ways that helices cross in observed structures? Is it compact? Is it clash? Those are all things that you can see in structures in the protein data bank. You can quantify that, and you can say, does it fit or not fit in what we see in the protein data bank. Okay, so this is should left some room here. This one. This one is sequence dependent terms, and this piece is sequence independent. Independent features. So basically, they brainstorm what looks like a protein, what doesn't look like a protein, what features do you see. So what? Secondary structure, packing is one thing they tested. They looked at 2 helices, helix, relative geometries, meaning crossing angles and distances. And they looked at where do you usually see helixes cross at? And there's certain patterns that are more or less variable. Another would be if you have, say, 5 strands, you could have 2 strands together and 3 strands elsewhere. So the strands could be distributed through different sheets. So it's the strand arrangement. So the probability of having the sheets that you observe could be some function of the number of strands and the number of sheets. How do those get distributed? And you can imagine, you can look through the protein data bank and say, when there's 6 strands, is it always 3 and 3, or is it 6 strands in one sheet, or is it 4 and 2? And how often does that happen? You can look at how often that happens in the protein data bank, and then you can look at how often it happens in your structures that come out of Monte Carlo, and choose the ones that seem more relevant. So these are a little bit, these are characterizing the secondary structure. Another feature is bumps. Something really obvious in the protein data bank is 2 atoms don't occupy the same space, they don't overlap. That's something we could look to a structure for. This is kind of like van der Waals. This is really just the repulsive part of van der Waals. In Rosetta, it's called VDW. And the centroid score, which you loaded on Monday, you loaded the full atom score. There's also a centroid score we'll do next week that has a VDW term. That's the bumps. It's the sum of Rij squared minus Rij, and this is the actual distance minus the tabulated distance that they would ever get between each other, the minimum distance between them. Those two squared, and we just sum those up. Say that's, if they get closer, any closer than the minimum distance, we're going to square the difference so that this be this quadratic penalty as they get close. And one more. It's the radius of gyration. So proteins tend to be compact. The way you can quantify that is with the RG. Radius of gyration, this is 2 over n minus 1. You're basically asking, if you look at it as a ball, how big is the ball? What's the radius? It's not a sphere, but if it was kind of like a sphere, about how big is that sphere? And you can do that by summing over each atom I, actually each alpha carbon, and each J greater than I, so you'll be looking at all IJ pairs of alpha carbons. And we look at Ri minus Rj. These are both vectors. And we look at the distance between those, square them, take the square root, do the combinatorics to account for it. That tells us in general how big the protein is. And in general, if you were to plot Rg versus, say, the number of proteins of a given size, say 80 to 100 amino acids, what is the Rg? They tend to fall in a certain range. And if you just start making configurations and folding fragments, what you would get, so this is in the PDB. If you just get random configurations, you'll get a lot of other things. So these are decoys. If you don't use an RG term and you just let it flop around, you get things that are all stretched out. No, that's good. So what we want to do is use an RG term to bring it, to encourage them to become smaller, and then eventually what you want is your decoys to have similar features to what you see in the protein data bank. Questions about that? So evaluating these on their own, you look at kind of these features, and are you reproducing the features in the protein data bank? Question, Lynn.
Isn't the decoys like a separate process from the scoring? So like when you create the decoys, do you like have a selection method to, I don't know.
Yeah,
dump
so
something?
we have that algorithm, and then after we make a new configuration, we calculate the score of the test compose, and then we do the delta E over KT thing. The delta E is based on these scores. So the scoring function is going to guide the search process, and if low energy means compact, then things will become more compact.
So as you change that delta E, I guess your decoys also gets closer to the PDB.
So I define delta E. So if this is the decoys where E does not include RG. If I just make decoys and I don't use any energy function at all, they're going to flop around. I'm going to make lots of different crazy things. Not many of them will be compact. I have to put in some kind of force to make them compact. So an RG term just explicitly says squish them together. What really I'm doing is emulating the attractive van der Waals that brings things together and the hydrogen bonds that tie the core together. But since I don't have all that in the centroid mode, RG is a proxy for that. We just squish together. So if the energy, the red here is where energy includes opposite natural log of the probability of seeing that particular RG plus other terms. So that the RG is, and we'll talk about the natural log tomorrow, Monday or next week, and we'll do that. So those are the sequence-independent, structure-based features. Then we can also look at, does the sequence fit on that structure? And we're out of time today, so we'll talk about that either on Monday or Wednesday.